as well? All
So
right, curious.
I also started out wanting to be a medical doctor because I grew up in a family that has a lot of people in the medical field. But I realized very quickly in my undergraduate, so like freshman and sophomore year, that I really just I wasn't as much interested in seeing patients as I was in figuring things out. I really like puzzle solving, and research is very much solving puzzles and figuring things out. So I think research is definitely the path for me. I'll be applying for my PhD directly in the fall, and I'm going to be staying on in my lab for the next year while I wait to figure out where I'm going to go.
That's
I'm
good.
also a senior, and I'll graduate in December. So I am at the Dr. Martin lab. He is a pediatrician. He specializes in gastroenterology. And I'm also part of the Bridges program. So I joined the Bridges program to really make sure before I applied for my PhD that I was really interested in research, because you think you know research and then you realize how much work it is when you're in it, and it can really tell you whether or not it's right for you. So my research is on the role of PISA-1, which is a stretch-activated ion channel, in the biomechanical stretch-induced growth of the small bowel. So just to begin, the small bowel, or the small intestine, is the part of the gastrointestinal tract that's directly connected to the stomach, and its main responsibility is to absorb your nutrients from food. The layers include this layer out here, which is called the muscularis, which what it sounds like, it has muscle cells, smooth muscle cells, but it also has the enteric nervous system, or the intestinal nervous system. So this layer is mainly responsible for the contraction and the movement of food throughout the GI tract. And then you have your epithelial layer, which includes these villi here, which are made up of absorptive cells, which are responsible for absorbing the actual nutrients from the food as it passes. And you also have these little circles here at the base of the villus, which are called the crypts. And the crypts house the multipotent intestinal stem cells of the gut. So the small intestine in humans is about 20 feet long in an adult, and that's in order to maximize the amount of surface area to fully absorb the nutrients and minerals that you need from your food. But my lab focuses on cases that actually have this length significantly reduced. And this results in something called short bowel syndrome, where you have maybe up to 60% of your intestine removed. This can happen due to conditions like gastroschisis, where a baby is born with their intestines developed outside of their body, which can actually result in this bowel going necrotic, or the tissue dying. There are also cases at birth or a few days after birth where the newborns can have what's called necrotizing enterocolitis, which is a bacterial infection. Question?
No, I think it was just accident. Go ahead.
Okay. Which is a bacterial infection that can cause the bowel wall to become inflamed and die. And for all of these cases, these children and these newborns require large portions of their intestine to be resected. And that results in an inability to really fully absorb their nutrients and minerals from their food. And it really delays their ability to grow at such a young age. This condition is also the leading cause of intestinal failure in pediatric patients. So there's only 3 options for these patients. The first one is what's called TPN, or parenteral nutrition, where nutrition is delivered through a Hickman line. This Hickman line is basically a catheter that goes from, it inserts into the chest and goes directly into the heart, and that's where the nutrition is delivered. Now, because this requires long-term IV access through their chest, 60% of these patients with SBS will end up getting sepsis episodes, so they get really bad blood infections because they have this chronic line. And because of the components within TPN and their limited movement ability in their gastrointestinal tract overall, these patients, I think it's like 40% of these patients will end up with damaged or failed livers. And then the next option for these patients that don't do well on TPN is what's called a step procedure, where it's an intestinal lengthening procedure that takes a dilated region of the bowel and makes these little zigzag cuts to increase the overall length, but it decreases the diameter of the intestine. So one side effect of this surgery is actually chronic obstructions or blockages in the intestine. And obviously, it comes with the risks that are inherent with surgeries, like infections and issues with sedation. And the success rate of this working is about 50% for patients. It really depends on the age of the patient when they finally get this procedure.
Okay, you have a question, Nicole, if you want to answer that one on the chat.
Yeah, let me figure out how to open it. Portia, how did you open the chat?
Where it says like share screen and stuff, there should be like a little yellow thing that pops up and you can click it. If not, I can read it to you.
So there's it depends on the condition that happens at birth to cause the need to have short bowel syndrome. Some babies are just born with it, and you can see that through x-rays. And other cases like the gastroschisis, it's more obvious at birth because their intestines are really outside their belly button. And then it's honestly, usually the reason they end up finding these issues is because the baby cannot handle food. The baby will be vomiting a lot. The baby will have chronic diarrhea. The baby will have stunted growth. Like a lot of these conditions have those symptoms, and those symptoms lead them to do the x-ray where they see like the necrotic bowel areas, or they see that it's significantly shorter.
I've got a quick question.
Mm-hmm.
Why does the IV drip with the nutrients have to be delivered directly to the heart? Like, couldn't it just go right into any like point of like into a vein somewhere else?
I mean, yeah, it could. Some of the babies actually, they try to do it through the they try to do what's called an NG or an NJ tube, where they like bypass the GI tract a little bit because they're still trying to force it to move to adapt. But I think the idea is to get it to the heart so it can be pumped out to the rest of the body faster. And most of these patients that are on this treatment run the TPM bag for like 12 hours a day.
So, but like being that it's going directly to the heart, doesn't that increase the risk of sepsis because it's going directly to the heart?
Any IV line increases the risk of sepsis because it's going straight into your blood.
Okay.
Yeah. So either way, they wouldn't be able to bypass that.
Gotcha.
Mm-hmm.
I have a question.
Yeah.
So you said sometimes with that, they get blood infections, correct?
Mm-hmm.
So what happens once they develop the blood infection? Like what, because that's the way they're being fed, like what the other option?
Honestly, these patients have a lot of reoccurring infections. These patients have to monitor their temperature daily, like probably twice at least a day. And if they get a temperature over 100, they have to go to the hospital. That's like their standard of care. And then once they're in the hospital, they get the antibiotic treatment. They test the blood cultures until the infection is gone, and they just send them home. But the issue with the chronic infections, it's also related to a high mortality, and it's related to a high risk of liver failure as well. So these patients that have these chronic issues with TPN will end up needing the last resort option, which is what I was going to talk about next, which is the possibility of an intestinal transplant.
Okay.
Yeah. And then an intestinal transplant, again, has its own risks like graft versus host disease and rejection.
Okay.
Okay. So this is just a quick video to show an example of a patient who has SBS. This patient had a volvulus at birth, which is when the intestine kind of flips on itself when the baby is developing in utero and causes a blockage of blood flow and passage, and it causes portions of the bowel to die.
Very
lion or a cheetah.
Riley came to rescue medicine after 3.5 months in a NICU down in Kansas City. Weren't going so well for her. She still wasn't able to eat, and so through a network of moms, I was able to get in contact with Dr. Mercer, and I called him on his cell phone, and he started the process of getting Riley up here.
And I realized, this is perfect. This is exactly who we need, the kind of patient who will thrive here. It became fairly obvious to us within a short period of time that we were going to have to do a little surgical intervention on Riley, just to change some things with her intestine. Step procedure is an operation whereby we take that big, stretched out, dilated intestine and we narrow it into a much smaller tube. It looks like kind of like a coil or a zigzag. And it allows us to take the big dilated piece of intestine and make it into a more narrow one that ends up being longer. And that really changes the way the intestines can squeeze and how the body's able to absorb nutrition. And so when children are struggling, like Bradley was, with vomiting and not being able to handle any feeds, you make this profound change to the intestine, and it's like a light switch is thrown. And within a week after surgery, the vomiting was all gone away, everything was turning around. And then it was just a matter of slowly and steadily advancing up on her calories and working on things by mouth.
By then, she'll be eating so much food.
Riley loves to eat anything we put in front of her. She loves cereals. She loves.
So again, this patient had the step procedure, and it was early enough to where her intestines could start adapting to this significant decrease in length, and she can start tolerating feeds. But there is a chance that while she can get some nutrition from feeds, she will still need to be on supplemented parenteral nutrition to get what she can't handle. Again, this only works in, 50% of cases, so this isn't always successful for these patients with SBS. So what my lab focuses on is looking at ways to improve intestinal lengthening procedures in order to help these patients with SBS adapt. Any more questions before I move on? No? Okay. So what we based our initial model off of was the people of Myanmar. In their culture, it is beautiful to have these elongated necks, and what they would do is they would slowly over their lifetime add these metal rings to slowly elongate their necks. So what we did is we developed a biodegradable capsule that had a spring inside that we could embed in the remaining intestine that could slowly stretch the intestine axially along a 14-day period, and that resulted in more than doubling the length of the intestine. And what we saw when we looked at a tissue level at what happened when we stretched the intestine, we see a significant thickening in this outer muscularis layer. So there's growth of the muscularis layer, and we see a significant growth in this epithelial layer as well. Specifically, when we looked at those crypts at the base of the villus here with those intestinal stem cells, you see significant growth and expansion of those intestinal stem cells as a response to the stretch. What we decided to do is develop another model that is more time efficient to study the mechanisms involved in what's happening in a lab setting. So we used a partial bowel obstruction model where we placed this little silicone band in the distal area of the intestine, which causes a partial blockage. And as food is being blocked up in here, it causes a circumferential stretch of the intestine. And in just 5 days of this model versus the 2 weeks at least of the previous model, we see the same effects on the histology. And what we were also able to do is we stained for an intestinal stem cell marker to really visualize what's happening within those crypts. And we see that in our unstretched state, we have them just at the base of this crypt. And when you stretch the intestine, you get significant expansion and hypertrophy of this red stain, signifying the growth of the intestinal stem cells. So what we're trying to figure out is what is the link between stretch and growth? What is happening at a molecular level that is causing this growth? And that brings us to piezo-1. So piezo-1 is a stretch-activated ion channel. When piezo-1 is stretched, it most often allows the entrance of calcium, and that effect has been linked to various cell processes and tissues that are commonly mechanically activated. For example, in vascular smooth muscle cells, so the smooth muscle cells that line your veins, under cases of hypertension, piezo-1 is activated by that pressure, pushing it outwards, and it has found that it induces the thickening of those vein walls where the vascular smooth muscle cells are. It has also been found to play a role in epithelial growth when stretched and stem cell migration. And lastly, it's expressed in the various layers of the GI tract, but its role there has not been well studied. But what has been well studied is the effects of calcium signaling. So calcium signaling is very important in the regulation of cell growth and gene regulation, as well as acting as a secondary messenger. A signal from outside the cell can bind to a channel, which allows it to open, and calcium can enter the cell, which can activate various processes and enzymes to do a myriad of things within the cell. Most notably is the cell growth and gene expression, and also it can trigger contraction. So our hypothesis is that the stretching of the intestine to induce growth is dependent on piezo-1 in the smooth muscle cells of the small intestine, and that piezo-1 is important to maintain normal intestinal contraction and calcium flux. So to begin with our methods, piezo-1 is embryonically, a piezo-1 knockout is embryonically lethal. So we can't just globally knock out piezo-1, because we wouldn't have a model to go off of. So what we have to do is we have to use an inducible knockout that we can induce when the animal, like in our case, mouse, is born. So we use what's called the Cree-Lock system. It's a complicated system, but basically it uses a promoter that's only activated in cell specific or cell type specific. So in our case, we use a promoter that's only active in smooth muscle cells. And we can treat the mouse with a chemical called tamoxifen, which activates this enzyme here to go cut at these two regions that we breed into the mice called lock sites that flank each side of our gene of interest. And then it cuts those sites and the gene is removed, excised from the genome. And we have effectively knocked out our piezo-1. In more detail, we breed these mice together to get our MYH11, which is a smooth muscle cell specific promoter, attached to our Cree, and we breed that with a mouse that has the piezo-1 sites flanked. And when we treat the mouse with tamoxifen, it activates the Cree to go cut at the two sites and knocks out our gene. If we don't treat with tamoxifen, if we treat with just whatever the tamoxifen was dissolved in, like the oil, or if we just treat it with water, it will still have piezo expression. So those are our control mice. We take these mice at about 5 weeks old, and we induce our partial bowel obstruction model in order to induce this radial stretch, and we look at the effects on post-op day 5. In our in vitro, so our culture, our cell culture model, we utilize the same mice, but we're able to take them at a younger age and strip the muscularis layer off the outer, we can strip the muscularis layer off the intestine, and we can seed it in culture to get this nice co-culture. So like the whole muscular system, we have the neurons, we have the smooth muscle cells, we have pretty much every cell type in the enteric nervous system, and we have serosal cells as well. And what we've developed is a way to grow these cells on a temperature-sensitive hydrogel. So it's a little piece of flexible, soft gel that responds to temperature. So when we seed the cells onto this gel, we take the gel at 37 degrees because it's in a shrunken state. And then when we decrease the temperature to 33 degrees, it expands and it stretches the cells that are seated on top of it. So it gives us a model to stretch the cells under the microscope. So, and then these cells, we also have it tagged so that when we're looking at under the microscope, whenever calcium comes into the cell will flash green. So to go into our animal model results, are there any questions before I move on?
Go ahead.
Okay. So to look at our animal model results, our graphs are a little confusing, but our circles are indicating our wild type mice and our triangles indicate our knockout mice, and the filled in symbols reflect if the mouse was obstructed. So when we're looking at the thickening of the outer muscularis layer in response to piezo being knocked out, we don't really see a difference between the wild type and the knockout. They both still increase in the same amount. However, the expansion of the crypts in the intestinal stem cells in our wild type group are significantly more than what we see in our knockout group. So we have a significant increase here, but the effect is blunted in our knockout group. And you can almost see it here on the histology, where these crypts are significantly longer compared to the original crypt size. But when you look at the knockout group compared, they are smaller. So our conclusions here are that piezo-1 and the smooth muscle cells of the muscularis mediate the crypt and stem cell expansion associated with stretch. Now to move on to our results in vitro in our culture model. This is looking at how piezo-1 would affect the contraction ability and the calcium influx happening on a cellular level. Our culture model is pretty cool because under the microscope, you can actually see the cells contracting. And we've done studies where we plate the cells on a membrane of gelatin, which can actually flap whenever the cells contract. The cells move the gelatin with them. So this is a time-lapse video of it happening. And like I previously said, calcium flux and calcium pretty much regulates contraction, especially in smooth muscle cells. So when we talk about calcium in the next coming slides and contraction, it's because the two are associated with each other. So at 37 degrees Celsius, so in our unstretched state, piezo-1 wild type cells have this nice spontaneous and synchronized calcium flashes. And you can see that here because all the peaks are lined up and they happen together. And there's not like too many of these long pauses. They're pretty equidistant. But then when you look at the knockout group at 37 degrees, so the same temperature, there's significantly decreased intensity of the flashes, first off, so the peaks aren't increasing as much. And they're not quite lined up, so they're not as synchronized as they are in the wild type group. And then when we decrease the temperature to 33 degrees Celsius and induce the stretch of the hydrogel and the stretch of the cells, our wild type group actually increases in frequency to double, and it still maintains relative synchronicity and spontaneity. It's really dramatic. But then when we induce the stretch of our knockout group on the hydrogel, there is almost no, it's completely arrhythmic and not spontaneous. This is all just background noise. So without piezo, there's no response to stretch, and at baseline, there is limited calcium flux occurring. And if it is occurring, it's irregular. We have also repeated these in human intestinal muscularis cell co-cultures, so similar cell culture to the mouse cells. But since we don't have a knockout model for this yet, we use chemical activators and inhibitors. So Yoda is a chemical activator of piezo-1, and GSM-TX4 is a chemical inhibitor of piezo-1. Looking at our control, we again have this nice spontaneous calcium flux occurring, and you can see it here indicated by this orange graph. And then when we activate piezo-1 by adding iota, we get a doubling in the frequency according to the graph, which you can see visually. And lastly, when you inhibit piezo function, you get a complete cessation of activity. You get no calcium flux occurring. So this data suggests that piezo is important for the maintenance of regular and rhythmic intestinal muscular cell contraction and calcium influx. Moving on to our next steps in our study, we've knocked it out in the muscularis, which we did it first in the muscularis just because it's more highly expressed in that layer, but it still has good expression levels within the epithelium. So we want to see what happens when we fully knock out piezo in the epithelium. And then lastly, when we can breed the mice correctly, we want to try to knock it out in both the epithelium and the muscularis.